So first off, thank you so much for this opportunity. It really means like a lot to me because I'm also gonna be pursuing medicine in my future, I hope for. So my name is Pratham Pai, and I'm here today to present my project with Dr. Samita Dal on the CRAS's impact on pancreatic adenocarcinoma using public genomic databases. Next slide. So before we go into the data, we kind of need to understand what cancer really comes from. So cancer is usually results from the overexpression or underexpression of two types of genes, oncogenes or tumor suppressor, and specifically oncogenes are genes that are with too much expression causes cancer, like CRAS, which is what I researched, or tumor suppressor, which too little expression can cause cancer, which is PTEN. And from here, you can see that on these graphs, CRAS, when at higher expression, red means tumors. Higher expression is associated with tumors, and lower expression for PTEN is associated with more tumors. So it's the opposite for both. I hope I said that correctly. All right, next slide. So the importance, the objective and the importance. So I use public genomic databases, GEPIA, TCGA, GTEx, and cBioPortal, and they all provide data about the function of the gene, how the gene is mutated, patterns and mutation rates, and gene comparisons. And the purpose of analyzing this was to provide scientists or researchers the ability to see patterns in the genes and understand like what causes these gene mutations and what happens across multiple patients suffering from these different cancers. And finally, it's important to consider the solutions that we can implement in our daily research that can allow patients struggling due to these diseases to find therapies and things that target these genes, either oncogenes or tumor suppressors. Uh, and again, I used genomic databases, GEPIA, GTEx, cBioPortal, and TCGA, and all these databases get their data from TCGA, which kind of records all the data from all the patients they see coming through the hospitals. And then all the patients are just put into distinct groups of data. So the first thing I analyzed during this internship was a box plot. And so box plots kind of show how different patient groups how different genes work in different patient groups. So when we look at this, red means the red box plot. The red box means tumor patients, and the gray box plot means normal, like patients who don't suffer from this cancer. And so when we look at it for the first time, we see that CRAS at higher expressions is associated with tumors, and at lower expressions does not have any tumors or the development of tumors. But when we look deeper into this, and that's what the goal of this internship was, to look deeper into data. And when we looked at this, we saw that when when you look at the like how stretched out that data is for the tumor group, you can see some of the tumor patients having tumors kind of overlap with those transcription rates of normal CRAS transcription rates. So someone going through the cancer might have a normal transcription rate of CRAS, but still go through that tumor, which made me believe that CRAS might be a fragile gene or might be more likely to get like a mutation in its gene. All right, so survival plots. There's two survival plots. One which looks at how the alterations of a gene affect survival and how transcription rates influence survival. And so the first graph shows the altered group in red and the unaltered group in blue. And when we look at this, the altered group has a much faster trend downward than the unaltered group, which makes us believe that CRAS, when altered, can cause a very increase, like a very fast decrease in survival from 100% to around 20% in just 55 months. And this is in pancreatic adenocarcinoma, which is one of the deadliest cancers, which makes sense because most patients unfortunately do not survive when they're diagnosed with this. And then the second graph shows that when at higher transcription rates, CRAS is more likely to cause a decrease in survival, while at lower rates, it's less likely to cause a decrease in survival, primarily because it's an oncogene. And the patient report kind of gives us a really in-depth understanding of the gene and how its mechanics. And when we look at this was a random patient that was chosen, um, a female with pancreatic cancer, and is unfortunately deceased into 8 months into diagnosis. And when we see this, we see that CRAS was the primary gene that affected this patient and caused this patient to get this cancer. And the mutation type was missense mutation, which is the change in one base in a gene. And the protein that was changed was G12D. And I kind of like I think this is very important to specify the protein, because when scientists look into the protein, they can kind of when they use gene technologies, they can target that one protein, and before it's even created, they can kind of alter it and change it so that it doesn't impact the patient later in their when it's being used. But that's something we need to go like further into, which I plan to do in the future. And then the gender pie chart, interestingly, no different gender is favored over the other in CRAS, and targets both genders the same. So gene comparison was one of the other things we were open to do for this internship, and I thought this was interesting because when you look at this, these two genes, CRAS and TP53, are both prominent in pancreatic adenocarcinoma. And when you see this, when we only look at tumors, we can see that TP53 requires around 4.5 transcription in order to cause a tumor. But when we look at CRAS, only 3 TPM is required in order to cause a cancer. So that kind of develops my it kind of develops my what I thought of CRAS in the beginning as a fragile gene, and that it can be influenced by environmental changes or like hereditary mutations very easily. And finally, the heat map and violin plot, I believe, is the last group of graphs. And these show us the transcription rates we need to be wary of in further studies and that cause these tumors. And so in the heat map, it measures at the log2 scale, and there's log2 and log10 scales, which are two different types of scales used to measure the transcription rates. And for CRAS in log2, it requires 4 TPM in order to initiate that development of tumors, or for the log10 scale, it requires 0.7 TPM to develop tumors. And that just kind of allows us to see before the development of a tumor in a patient, if they suspect a tumor, like if they suspect being diagnosed with that cancer, they can see if they do they have that transcription rate, and is it possible that they will get that cancer. Oh, and the mutation plot. So the mutation plot, it analyzed 3 different types of mutations: missense, inframe, and no mutation. And so missense mutations, which we saw in the beginning, they are mutations that are caused by one change in one base. Inframe mutations are changes in multiple insertions and deletions in the gene that cause like multiple frame shifts in the gene. And no mutation just means that the body just uses that gene more than it should usually be used, and so that causes the development of tumors. And from our studies, we saw that more patients from this study were likely have the missense mutation in their CRAS mutation. And so in conclusion, we found that CRAS is an oncogene that is mutated due to the CRAS mutation, due to a CRAS mutation, a missense mutation, and higher expression of the G12D protein can cause a cancer to break out in the body. We also found that CRAS is much more of a fragile gene than we previously might have thought. And studying these genes allow us to find patterns in the expression of each individual gene and protein that allow researchers to target these genes and try to lower or increase the expression rates for both oncogene and tumor suppressors. Thank you. That's yeah.
Wonderfully made presentation, Pratham. Thank you so much for presenting. So if anybody has any questions, so awesome work, Pratham. It comes from Richa.